Hi everyone. This video is going to look at the role of cannabis and memory. And so this is going to kind of expand on what we talked about a little bit in the emotion lecture, where we looked at the ability of the endocannabinoid system to control memory extinction. Here we're going to look at the role of cannabis, the endocannabinoid system, in learning in general. So the ability to remember something the first time. So this was what we looked at before, and you probably remember this. Well, maybe you don't remember this if you were doing some let's call it experiential learning, but I'm going to recap it a little bit here. So we were looking at learning extinction, and we had a mouse in a maze. And if we put the mouse in the maze ran around until it found the cheese or the reward. And then if we put the cheese in a different corner of the maze and then put the mouse in, it would immediately go where it found the cheese the first time. And when it saw that it wasn't there, it would run around until it found where the cheese was. And it didn't matter whether this mouse was a control mouse or a mouse that had the CB1 receptor knocked out. The difference came in the third trial. So in this case, the cheese was in the back right corner again. It was where it was in trial 2. The control mouse remembers that the last time I did this, it was in the back right corner. And so it goes straight to the back right corner. However, the mouse lacking the CB1 receptor with a defective endocannabinoid system still thinks the cheese should be in the back left corner where it was the first time. And so it goes to the back left corner and then runs around until it finds it, just like it did in trial 2. And no matter how many times you put this mouse in, it still thinks the cheese should be where it was the first time. So how does the endocannabinoid system play a role in just memory alone? And so we're going to start with short-term memory. And so in order to talk about this, we need to understand how these short-term memory experiments work. So in this situation, we have 3 lights. We have a central light and then 2 lights on the side, and each light on the side has a lever associated with it. The light in the middle can flash one of 2 colors. In this case, it can flash either blue or orange. The mouse sees what color that first bulb flashes, and then, in this case, it's flashing blue. We then light the other 2 lights up. One of them is blue, one of them is orange. If the mouse pushes the lever for the light bulb color that matches the first light bulb, it gets a reward. If it pushes the lever that does not match, it gets nothing. And so if you do this enough times, the mouse realizes what's going on. It sees a flash and then realizes, I need to push the lever with the same color. Okay, so that is the training. We've trained these mice on how to complete this experiment. Then we do the experiment. So now we're going to take that mouse and we are going to give them a CB1 agonist and see what happens. So in this situation, we flash the center light blue again. We then show them the 2 colored options, but the CB1 agonist mouse can't remember what color the first light flashed. And so it just picks one. Sometimes it's right, sometimes it's wrong. It may even know that it needs to pick the one that matches, because that is long-term memory that occurred before we gave them the agonist. But now it can't remember in the seconds between the first light flashing and the other 2 lighting up what was the first one. And so it just takes a guess. Similarly, we can measure this looking at how familiar our mouse is with an object. When you show a rodent a new object, its instinct is to investigate that object. And so if you give it 2 objects, one of which it has seen before and one of which is new, it's going to spend more time looking at the new one and less time at the old one. And so here, we have 2 sets of mice, a control set of mice and mice that are given THC, and we're going to put the same set of objects into both cages. We have a round object and a square object. And the first time that you put these objects into the cage, the control mouse spends 10 seconds with each, the THC mouse spends 10 seconds with each. For both the control and the THC, these objects are new. They're going to spend a fair amount of time with them, and they spend equal amount of time between them because they were new. Now we're going to remove the square object and replace it with a triangle object. So now we have an old, familiar round object and a new triangle object. For the control mouse, the circle object was familiar, so it only spent 2 seconds with it this time, and then 10 seconds with the triangle one. The THC mouse, though, can't remember that it has already seen the round object, and so it spends 10 seconds with the round one, 10 seconds with the triangle one. And so it is very clear that in this case, THC is affecting short-term memory. If we take a mouse and we give it THC chronically, and then withdraw the THC, we still see this effect. However, it's unclear is the effect sticking around after the THC has been withdrawn, meaning that the THC, or the chronic THC, resulted in long-term changes in brain anatomy, or not, probably not anatomy, brain physiology and function, or, remember we talked about how THC sticks around in the body for a very long time. And so which one is causing this, we don't know. Functionally, it doesn't really matter. It's very clear that long-term THC exposure causes long-term, short-term memory loss. We can also look at the role of THC in spatial memory. So here we're putting a mouse in an apparatus that has essentially a chamber in the center, and then there are hallways radiating off of that center. At the end of one of those hallways is a reward, such as a piece of cheese. The mouse, when it's in the center here, cannot see the cheese at the end of the hallway. The only way for it to find the cheese is to investigate the different hallways until it finds it. And so the way you or I would do this is the same way a mouse is going to do it. It's going to use process of elimination to start picking halls until it finds where the cheese is. But it's able to remember which halls it has already checked. And so every time it checks one, it then checks another one that it hasn't checked. That's process of elimination, right? It's eliminating the ones it has already checked. However, a mouse that is given THC can't remember which hallways it has already checked. So it may check the same hallway 5 times before it checks the one that the cheese is in. So we see that here. It's checking the same hallways multiple times. It checked this one twice, and I think it was this one once before it found the reward. So this is all short-term memory, but there are different forms of short-term memory, right? We have spatial memory, we had object memory, and then we had kind of visual, light, or pattern memory. So you're probably thinking, well, if CB1 agonists block memory, then what about CB1 antagonists? With CB1 antagonists, called an increase of memory. In this case, it would kind of be like the movie Limitless, where there was a pill that essentially allowed the actor or the character to access what they called 100% of their brain, and they became much, much smarter than everyone else. So is a CB1 antagonist going to do this? Lucky for us, CB1 antagonist exists. And actually, it has already gone through clinical trials. We'll see this compound later on, but this compound is called rimonabant. Like I said, it has already gone through clinical trials, though not for memory. If we give rimonabant to mice, it is obvious that it is increasing olfactory and spatial memory. If we have CB1 null mice, they have increased object memory. Now, just because I didn't include object memory in the rimonabant, and I didn't include olfactory and spatial memory with the CB1 null mice, doesn't mean that they don't have increases there also. That just means we haven't tested it yet. But it's very clear that CB1 antagonists or knocking out the CB1 receptor increase memory. However, oddly enough, if we go back to the experiment with the lights and the levers, neither one of them affect that. And so that's sort of confusing, right? So what's going on here? The best explanation is that there is a time difference, a scale here is different between the types of experiments. With the light and lever experiment, the first light goes, and then just a couple seconds later, the other ones light up. In the other experiments, there's minutes, hours, days between. So in both situations, it is short-term memory. But perhaps there's a difference and an effect on short-term memory versus long-term short-term memory. I hope I didn't screw you up when I said that. I hope that makes sense. So what goes on when you give these agonists to a person that's causing these effects? We've already seen that CB1 agonists and antagonists as well have their effect primarily in the hippocampus. And it appears that CB1 agonists have essentially the same effect in the hippocampus as a hippocampal lesion. And so it is more or less preventing the hippocampus from working properly, and that region of the brain is very, very, very important in short-term memory. If we look at agonists, they decrease glutamate release. That's something we know, right? That's not specific to this lecture. Cannabinoid receptor agonists block glutamate release. But glutamate release is required for synapse formation. And our memory works through the formation of synapse. My synapses aren't working properly. Glutamate release is required for synapse formation. I'm just going to move on. If we block that synapse formation, we are going to block memory. Somehow, this blockage of synapse formation appears to be through COX-2. But we have plenty of cyclooxygenase or COX inhibitors, including aspirin. COX inhibitors block this anti-memory effect of CB1 agonists. So what about in humans? It is well known, and has been known for a very long time, that cannabis use causes short-term memory loss. This is portrayed very, very frequently in TV shows and movies. I remember an episode of The Big Bang Theory where I think they stole some cookies or brownies from some campers nearby, didn't realize there was cannabis in them. And so the whole time they're out there, they wanted to eat something, and after they've cleared out the cooler, they all of a sudden think, well, maybe there's something in the cooler. And then over and over and over again, they keep checking to see if there's anything in the cooler, even though they've already cleaned it out. It's known that CBD blocks this short-term memory loss. And so again, current street cannabis has high THC, low CBD. The older strains had less effect on short-term memory. How long this effect lasts is not quite clear. And we know it lasts weeks. But again, is it the effects lasting weeks, or is it the cannabis lasting weeks? We don't quite know yet. Beyond a couple of weeks, we don't know if cannabis has a long-term effect on short-term memory. partly because there's a lot of things going on here. If we look at recreational cannabis users, they're going to be using different strains of cannabis. So that's a confounding variable. We're going to have issues with socioeconomic status. Perhaps people using cannabis fall into, or are more likely to fall into a particular SES category. And if we look at memory or learning amongst SES categories, we're going to see differences. And so that's a confounding variable. We also know that people that use cannabis for one reason or another, and we'll talk about this later on, are more likely to use other drugs. And so if they're using other drugs and suffering from memory loss, no, we just have too many variables. And which one is causing this? So it's very, very hard to figure out. I am not a statistician. I'm glad I'm not the one who has to do it. So that is that. Hope you found it interesting.